Hi everyone, this is Andrei-Marius Avram. I am the first author of this work entitled Species Entity Recognition using a Neural Inhibitory Mechanism, and we use this work in order to participate at the first task of the LivingNER competition. The motivation behind our work is quite simple. We strongly believe that clinical NLP is a valuable research field that helps us to automatically understand and analyze this knowledge that is written in these notes. And as a result, we choose to participate at the first task of the LivingNER competition, which is part of a bigger conference, namely the IberLEF 2020. And this task focuses on extracting name entity recognitions for species mentions and also for entity linking, providing an exhaustively annotated corpus for Spanish clinical case reports, which other researchers can use. And the main approach that we used in our work was to employ the newly introduced lateral inhibition layer, a layer that is inspired from the inhibitory mechanism of our neurons, where basically if one neuron strongly activates, then it inhibits the activation of its neighboring neurons, which usually results in contrast or sharpness increases in things like the visual or the tactile response of the human body. And more specifically for our work, we believe that the lateral inhibition layer may help us in better identifying which regions of the input are more important for predicting the name entities in the text. So basically in our system we tokenize the text and then extracted some contextualized embeddings using a pre-trained model, namely XLM-RoBERTa. And then on top of those contextualized embeddings we employ the lateral inhibition layer in order to put more emphasis on the features that are more important for identifying the desired name entities. And also we experimented with various configurations where we remove modules at random like the lateral inhibition system, and we also experimented with an ensemble system that is composed of all those systems that we experimented with. In addition, we also employed a data augmentation pipeline that consisted in translating into English the sentences that were between the name entities and then translating back those English sentences into Spanish. So in the end we obtain some kind of augmented text that preserves the semantics and the meanings of the original text. Here we have the results for the development set, and as you can see we experimented with 5 systems, out of which we have one that is the ensemble system, and out of all these systems the ensemble one obtained the highest total F1 score with 95.84%. And during the testing phase we sent again all these 5 systems for evaluation, and this time the highest F1 score was not obtained by the ensemble system but by the XLM-RoBERTa with the lateral inhibition layer, and it obtained an F1 score of 95.03%. And we can also observe that the translation augmentation did not improve the results for neither the XLM-RoBERTa without the lateral inhibition layer nor for the XLM-RoBERTa with the lateral inhibition layer, and we believe that the cause of this decrease in performance was produced by the fact that this augmentation introduced additional noise, which made the overall system more brittle to new examples. In conclusion, this presentation describes our system that was used to participate at the first task of the LivingNER competition, and for this task we employed the XLM-RoBERTa pre-trained model together with the lateral inhibition layer, and we also experimented with things like translation augmentation and with ensemble systems. The highest F1 score was obtained by XLM-RoBERTa with a lateral inhibition layer, obtaining an F1 of 95.03%, and this result put us at the second place on the leaderboard for this task. Finally, I would like to thank you a lot for your attention, and if you have any questions regarding this work, please contact me directly at this email, andre.avram@rakai.ro.